Welcome to this video on the principle of whole genome bisulfite sequencing, or WGBS, a powerful technique for unlocking the epigenome at single-base resolution. This section provides an introduction to WGBS, exploring the ultimate methodology for comprehensive DNA methylation profiling. What is WGBS? It is a global methylation mapping technique that combines sodium bisulfite treatment with next-generation sequencing. This enables the interrogation of DNA methylation patterns across the entire genomic landscape at single nucleotide resolution. WGBS provides unbiased genomic coverage. In the human genome, this involves evaluating the methylation status of approximately 28 million CpG dinucleotides, ensuring that no regulatory region or enhancer escapes epigenetic analysis. The three core pillars of WGBS are bisulfite treatment, NGS sequencing, and bioinformatics. These steps work together to convert DNA, sequence fragments, and align reads to pinpoint methylation loci. Next, let's delve into the chemistry behind the process, specifically understanding the mechanism of sodium bisulfite conversion. The core of WGBS relies on the difference in how unmethylated and methylated cytosines react to bisulfite. Unmethylated cytosine converts to uracil and reads as thymine after PCR, while methylated cytosine is protected and remains cytosine. This chemical alteration transforms the genetic code. WGBS translates unreadable methylation states into distinct, sequenceable genetic variations, which is key for epigenetic analysis. WGBS is widely recognized as the gold standard for DNA methylation profiling. It offers unparalleled single-base resolution across the entire genome, as highlighted by the Epigenetics consensus. We now move to the laboratory workflow, detailing the journey from a biological sample to a sequenceable epigenome library. The WGBS protocol involves four main steps: extraction of high molecular weight DNA, library prep with fragmentation and adapter ligation, bisulfite conversion, and final PCR amplification. Library preparation involves fragmentation and ligation of specialized methylated adapters. These adapters are critical because they protect indexing sequences from bisulfite conversion, which would otherwise convert cytosines to uracil. Critical treatment parameters include harsh conversion conditions that cause DNA degradation, the need for over 99% conversion efficiency, rigorous purification to remove inhibitory salts, and the selection of specific uracil-reading polymerases for amplification. The WGBS platform overview starts with sample input, then uses high-throughput sequencers like the Illumina NovaSeq, finally producing massive FASTQ files as data output for computational analysis. The final major step is bioinformatics and data analysis, which focuses on decoding the bisulfite-converted sequence to accurately map methylation patterns. Aligning reads after bisulfite conversion is challenging because most cytosines have become thymines, reducing the genome to a 3-base alphabet. Specialized aligners, like Bismarck, are used to map these complex reads. Methylation in mammalian genomes predominantly occurs at CpG dinucleotides, making up about 75% of methylation contexts. Non-CpG methylation is rare, though slightly more prevalent in embryonic stem cells and brain tissues. Mapping efficiency in WGBS is lower, typically 60 to 70%, due to reduced sequence complexity after bisulfite conversion. In contrast, standard whole genome sequencing is usually over 90%. To confidently call a site as methylated or unmethylated, a minimum sequencing depth of 30x is recommended. This high coverage depth is necessary for accurate methylation calling across alleles, requiring substantial sequencing power. We now transition to the applications and comparisons section, exploring why WGBS is often the preferred choice over other methodologies. Applications of WGBS include cancer profiling, tracking methylome remodeling during development and cell differentiation, and discovering novel epigenetic biomarkers for personalized medicine. This table compares WGBS with other technologies. WGBS provides the highest resolution and coverage, making it the most comprehensive method for whole genome methylation analysis.